I'm Daryl Helm. Our first speaker today is Christian Peter Klingenberg. Before he speaks, I would just like to remind you we're having this dinner this evening over at Clare College. Clare College has a great reputation for its kitchen. Yeah. And on the other hand, we're not actually going to the main part. We're going to a place called Gillespie Center Memorial Court at Clare College. Okay, so there are maps here. Okay, it might not be on your Google, but there's a little X on where it is. So I'll just put it on the table on the back in case you want to pick it up.
Okay, good morning. This is going to be slightly different from what you've heard in the last couple of days because I guess I'm the token biologist at this conference. So I'm going to present the perspective from the view of an evolutionary biologist, someone who studies the evolution of shapes and sort of the developmental and genetic basis of that, using a framework that biologists call geometric morphometrics, that is basically statistical shape analysis. I think this fits very well with this meeting on growth and form. So we've heard some tributes to Darcy Thompson already. Darcy Thompson was sort of an interesting a character sitting between disciplines, but definitely as a biologist, I think I have a claim of a large chunk of him for what he did in biology and how he inspired a subsequent work in biology. Certainly what we're calling morphometrics, the biological side of shape analysis, is hard to think without Darcy Thompson's contribution. Some of the techniques we're currently using are actually to emulate the transformation grids that Darcy Thompson published 100 years ago. For data, for the things we're studying, I'm gonna stick to landmarks. So it's a more sparse description of shape. For the simple reason that landmarks are the only the only things where we actually know exactly about correspondence between different forms, and particularly if we're looking at larger scale evolution of shapes, that does matter. I've had discussions here already with a few of you about this problem of correspondence. I'm eager to learn, but so far I haven't quite heard the key thing that makes me stop worrying about the correspondence. Correspondence is very important and is key to the results we're getting from analyses. Therefore, for the moment, until I'm convinced otherwise, I'm going to stick to landmarks where correspondence is clear. We can justify anatomically why we're convinced that this point in one species is the same as this point in another species. So landmark points, they're points that can be located precisely in every specimen and correspond in a one-to-one manner. Underneath everything I'm going to talk about is Kendall shape space. That the approach has been initiated here in Cambridge by David Kendall, neighborhood 40 years ago. That is a solid foundation for analyses. The nice thing about biological data is that even what we as biologists think large-scale comparisons usually are in a sufficiently restricted range of shapes so that the tangent approximation to Kendall's shape space is good enough. Here's a classical example from back in about 2000, Les Marcus and colleagues, a comparison of skull shapes across all the mammals, with the correlation here for the actual procrustes distance, shape distance between individual specimens and the tangent distance, and it's a very, very close correlation. It does matter if you squint. You may see that there are actually two regression lines here in the plot. One is with and one is without the dolphin, which is sort of the weirdest of the skulls in there. So outliers do matter a little bit, but regardless, the tangent approximation is quite good for the sort of scale that we're usually encountering in even large-scale biological studies. As biologists, so we're probably using we're less adventurous than you here in terms of the choice of method. So a lot of us use a relatively standard set of frameworks for thinking about shapes, analyzing shapes. Where we're doing a lot then is in terms of the structure of the data, and so using this framework of the tangent space to Kendall shape space, within that framework we're doing a variety of things depending on the data structure, depending on the study designs, and that's what I'm going to talk about for the rest of this presentation. I'm going to run through a number of examples, different data structures, different contexts, and then also different types of analyses.
Excuse me, what you call tangent distance is the geodesic distance?
No, it's the Euclidean distance in the tangent space. So the procrustes distance here, that's the geodesic distance on Kendall shape space. And so that's projected into tangent space to Kendall shape space at tangent point, the average mean shape, and then tangent distance is Euclidean distance in the tangent space. First example, human's best friend, domestic dogs. You have a huge variety of head shapes. As a biologist, as an evolutionary biologist, the particularly nice things about the evolution of dogs is that the common ancestor, the wolf, is still around for comparison, and that variety also applies to skulls. Here we have the wolf, and we have different breeds of dogs here. A huge variety, and in those, my former postdoc, Abby Drake, brought a data set to my lab using initially 65 landmarks. Once we ran the analysis, we reduced it to 50 landmarks on the skulls. So landmarks at the sutures between different bones in the skull, and also around the teeth here. Abby had a data set of in the neighborhood of nearly 700 domestic dogs. For comparison, somewhat over 250 wolves, so we have the actual ancestor there. And the closest wild relatives, the coyote and the golden jackal. And as a comparison for natural evolution, we chose to include the order Carnivora. That's the larger group within which dogs have evolved. That includes things like bears and cats and seals and weasels, etc.
So in this picture, the top one is the wolf?
Both of those are two views of the same skull of a St. Bernard dog.
The same dog?
A St. Bernard dog. One skull, top view, bottom view. Sort of an overview. This is looking at the shape tangent space, or the direction with most variation, principal component analysis. Looking at the distribution of those different groups in shape tangent space. Up here in the first versus second principal component, first versus third, second versus third. So we see the domestic dogs are hugely variable here in green. Much more variable than the wolf, the coyote, or the jackal. So a hugely expanded amount of variation by comparison to natural species. Comparable in magnitude of the variation to the variation across the carnivora. So the carnivora, that's something like 60 million years of natural evolution. The domestic dogs, that's the effect of a few thousand years, mostly a few hundred years of artificial breeding by humans. Here in the plot of second versus third principal component, which is sort of a lucky angle to look from, we actually see that most of the variation in the domestic dogs is outside of most of the variation in the carnivora. So a lot of shapes, skull shapes of dogs are novel by comparison to what's ever been around in the carnivora. We have the shape changes that go along with those axes. So this first axis with the most variation goes from a very short skull, short broad skull, to a long and slender skull. So on the left here we have things like pug, Pekingese, bulldog, English bulldogs, etc. On the right we have collie, borzoi, breeds with long and slender skulls. We can quantify the amount of variation for comparisons. So the amount of variation in there, either using the total variance of the shape across all dimensions, or using the volume of the convex hull, so the amount of space that they're occupying. In either case, what comes out is that, again, sort of inconsistent with what we've seen before, domestic dogs, whether we look at them as individuals or as breed averages, are much more variable than any of the natural species, wolf, coyote, jackal, and are comparable in magnitude of variation almost with the order Carnivora as a whole.
Sorry, what's Procristus variance? Can you
Procristus variance,
what you're
okay, that's the total variance, the sum of variances across all the dimensions of shape tangent space. Or mean squared distance of the individuals or breed averages from the respective mean. The volume of the convex hull in the shape tangent space, volume of the convex hull, limited to 3 dimensions there, because we have many dimensions with very little variation in there, so rounding errors get important. Yes?
What species have you put the tangent space? At dog?
At the overall average in the middle there, that is sort of minimizing distortions of the tangent projection for sort of everything.
Okay.
And again, the scale of variation is comparable to what we've seen with.
What's the nearest species to this general average?
The nearest species, it would be a domestic dog.
Ah, really?
So and it's.
Sorry, which domestic dog?
I honestly don't know. So it's something with.
Well, it should be at 0, 0.
Yes, it oh, sorry. It's at 0, 0, so it's the 0.00. So it's something with a skull a little short, not as slender as a wolf. So, yeah, beagle, dachshund thing. Unremarkable domestic dog.
So the question for statistics, so I imagine it's easy to access domestic dog, but for wolf, how do you get them, and what region of can you get access to?
All the wolves were from Alaska.
Africa? Yeah.
Alaska.
Oh, Alaska.
Where wolves are shot on an industrial scale and
Yes.
go into museum collections. Domestic dogs are actually, well, domestic dogs are unusual by comparison to other domestic species in that there's one collection in Bern in Switzerland that has by now about 3,000 skulls of purebred domestic dogs. For any other domesticated species, that's not nearly as easy. That's why there's much more research done on domestic dogs than any other kind of domesticated species. So it really depends on availability of collections, and there's a bit of chance there. But in terms of where the wolves are coming from, it would matter very little. So the North American wolves versus European versus East Asian are quite similar. Okay, we've actually then asked a little bit to, hey, how did this huge variety in domestic dogs come about? What are the causal factors? Is it selection for bizarreness, or is it relaxation for selection so that domestic dogs no longer have to run after prey and chew tough stuff? That's actually quite hard to address. The closest that we managed to come is to compare different groups of domestic dogs, pet breeds and different kinds of working breeds, so guard dogs, different kinds of hunting dogs, herding dogs, northern breeds, sort of sled dogs, and similar things. And it sort of comes out that the pet dogs on the left, companion dogs, are more variable than any other of the single other groups, often about, depending on the measure of variation you use in terms of volume of the convex hull, the amount of space they're taking up, they're comparable to all the other groups together. So the breeding for bizarreness is definitely supported there. and also that there's more tolerance for pet dogs to have a skull shape that may interfere with functions like breathing, etc.
What is bizarreness?
Bizarreness is just about breeding for unusual kinds of shapes, even though it may be non-functional. So we have English bulldogs now have such short skulls so that the soft palate gets in the way of the airways. That would be totally useless for something like a herding dog that has to run around a lot. So it's dysfunctional morphology that people have selected for. Okay.
Question.
Yep.
Is there a correlation between bizarreness and life expectancy of a dog?
To some extent, yes, but it's complicated. One of the key points is size. Big dogs are short-lived. Smaller dogs tend to be longer-lived. The biological basis of that is rather unclear to me. It's unlike other animals. It probably is something of genetic baggage that they have. Yes.
I just read that elephants have very good genetic defense against cancer, and therefore they could become so large. Is the reason for the short-livedness of large dogs, is it cancer?
Not only. Also, but not only. Okay. Overall, so I've mentioned about the structure of data, so here's a summary of sort of general scheme of what we often encounter in biology. So, often including what I mentioned, we have a number of different species and different species of biological organisms. They have evolved, they're the result of an evolutionary history, so they're linked to each other by a phylogeny which we can approximate as a tree-like structure. It's actually not properly tree-like, but network approaches in phylogeny are just about only just starting, and again that would be the challenge for the mathematical side here, to think about how to treat those. Trees are reasonably feasible, and it's possible with a reasonable amount of reasonably accurately to think of the relatedness about between different species as being tree-like. Within each taxon, each species, often we have different growth stages, baby, juvenile, adult, and we can collect data across the different growth stages to look at growth within the different species, and then we may have other information, usually within a particular growth stage of a particular species, about functional aspects, about developmental interactions, usually that from analysis of left-right asymmetry. We may have genetic information about that. We may have, depending on experimental setups, information about environmental sources of variation that may come into play.
So, excuse me, it seems to me that every one of those diagrams is essentially the same.
Yes.
As the other one.
So within each of those, we can analyze the structure of variation within each species, each growth stage,
Can you explain these diagrams? I don't understand.
Okay.
What's the connection between the lower right on the left and the upper left on the right? That connection from the corners. What does that mean? From the lower right on the left in one of those pairs.
Yep.
On the lower left, there's a connection to the upper. I mean, the lower right is the upper left. Are these identified?
Don't worry too much about that. The diagrams I've stolen from myself elsewhere to sort of a cartoon of standing for the structure of variation that we can look at within a sample. It's strictly about in biologist talk, modules, so complexes of traits that covary with each other tightly and are relatively separate from others. It stands really what I meant it to stand here is each of these pairs stands for the structure of variation in a sample, and the samples can be composite of we have them across different species for each stage. We can look at within each stage we can compare the different species to get differences that have evolved have arisen by evolutionary divergence of species. We can look across the growth from juvenile to adult within species to look at processes of growth, changes that come about by growth, and depending on experimental circumstances within a given species, a given growth stage, depending on what extra information we have, we may have those contexts in terms of function, in terms of developmental processes, in terms of genetic determination as well. This is just about general cartoons to stand for the structure of the data, the structure of variation in the data.
What is modularity?
That's another seminar. I'm afraid. I'll be happy to. I'll be happy to talk to you over coffee. With this multi-level approach, the ability to look at variation at these different levels. The interesting thing for the biologist is that the connections between levels relate to biological processes. So the connection, for instance, between genetic variation, an example, and evolutionary change, that is processes that are understood theoretically and can be quantified in terms of what happens change in response to selection, neutral drift, etc. The link between function and evolutionary change, again, is selection based on performance in a fitness-related functional context. So for that reason, as biologists, we're interested in comparing variation at different levels. I'm going to go through an example quickly to show what is sort of there in actually many data sets. The example is leaf shape in 20 species of this plant potentilla, which you can find around here. We've had here the species averages again in the shape tangent space, is the result of a principal component analysis. First versus second principal component, plotted species averages. The phylogeny is projected in, so you can go here to the point of the root there, that's the common ancestor. The lines then are sort of the best available estimates of historical changes, evolutionary changes that might have happened there. Here we have the eigenvalues, so the variation, shape variation in those leaves is concentrated a lot in the first few dimensions. There's many dimensions of the shape tangent space that have hardly any variation at all. So really everything is concentrated in about 3 or 4 dimensions and then fizzling off very rapidly. Shape changes associated with those directions. First principal component is sort of the relative length of the petiole versus the size of the leaflets. The second principal component in the vertical axis here, it stands for variation in the shapes of individual leaflets. Petiole length is more or less constant. This data set, so we have the data from 20 different species, for each species 10 different leaves, and then also the leaves are left and right symmetric, so we can look at 3 levels of variation. The one we've seen already, that's just a copy of what we've seen before, so we can look at the variation between species averages, in this case also taking independent contrasts to take into account the phylogenetic nature of the data, essentially a generalized linear model that takes into account the interdependence of the different species based on the phylogeny. Then we have variation within taxa, so the pooled within PCF, the pooled within taxon covariance matrix. So that's standing for individual variation among leaves within each species. And then finally, we also can look at the left-right asymmetry, variation within leaves that comes essentially partly from the inherent noise in developmental processes, but also partly from phenotypic plasticity, so responses to local environmental heterogeneity. We see, if we look at the shape changes here, we also can, if we compare the principal components, the eigenvectors as directions in shape space, we can find that, or if you run some models for more formal comparison of the PCs, we can see a similarity here, which relates actually to population genetic theory, where there are predictions that under a model of random evolution by random drift, we would have the same pattern within species versus among species, that conservation. So one theme, a lot of biological data sets are sort of multi-level like this, and we can ask a series of different questions based on the structure of the data. This is another topic that sort of goes back to the time of Darcy Thompson. It's actually a little later. The original of that appeared I think, in a Darcy Thompson sort of commemorative volume, already a symposium about on growth and form in the 1940s, looking at allometry. So we have changes in shape associated with with changes in size. Here, humans. Here on the left, there are some fetuses, a newborn baby, an adult. So we have changes in the proportions that go along with overall changes in overall size. In the context of statistical shape analysis, we can analyze that in terms of a regression, multivariate regression of shape on size. And again, we can do that at a number of different levels. So for the potentilla example I've just shown, we can do it at the level of species averages. So we would have then the changes of leaf shape averages, shape that divergent evolution of shape in response to divergent evolution of size, or we have individual variation within species, and again a pool within taxon analysis. So we look at the covariation of the deviations from species averages in terms of size and shape. I mentioned already the phylogenetic structure. That is an important aspect of many biological data sets. Here, a slightly larger example, flywings, about 58 species. Here, again, the projection into shape space of the phylogeny, displaying how the different species are spread around shape space. We can look at the association between size and shape through evolutionary. So does shape evolve together with size as across the phylogeny? But then we also can, and this is the final thing I'm going to start, we also can look at different species. How do the allometries change there? How do the allometries themselves evolve? So how do the relationships between size and shape evolve? So here we've got an example with those flower shapes. There's one from with size associated. So we have a smaller flower here, we have a larger flower here. The proportions don't change, the shape is the same. Or we can have these tendencies here, allometries, where with increasing size the flower gets more elongate, or in reverse, the flower gets broader and wider. For each of those species, we can run a regression of shape and size, and the resulting regression vector provides a description of the allometry of that association between size and shape. We can then do an ordination of that. In this particular case, they all would align nicely along one single axis. In practice, those patterns may be more complex. So this is what we've done with some colleagues from Argentina in a group of flowers called Loasaceae. They've got fairly complex flowers, most of which are pollinated by bees. Some of them are have evolved to be pollinated by hummingbirds, and one rather bizarre species is pollinated by small rodents, sort of mice. And if we do the ordination of the allometries here, so we have the individual allometries for the petals here, left diagram, small size, right diagram, large size for each species in this, on the right side here. So for most species we see from left to right that with a small, for the small sizes there's a kink here in the line down here, and this straightens out with larger size, so as the flower opens the base of the petal straightens. That is for most, not quite all, so for some this kink is not quite straightening out, so there's variation in that. And there are other features of variation as well. Here for the staminode complex, for most species we see this, the nectar scale, the lower part here, overgrows the staminodes themselves, and again that happens to variable degrees. We see then in the ordination of those allometric patterns there's a good deal of variation, and if you look where the black dots are, hummingbird pollinated species. This gray dot here, that's the mouse pollinated species. So for the petals, hummingbird and mouse pollinated species seem to have similar allometries, whereas the bee pollinated species have much more divergent patterns. For the staminode complex, the two hummingbird pollinated species are here sitting on one extreme, but now the mouse pollinated one is a long way away from them, and again the bee pollinated species spread around. So there's a number of different analyses we can do in response to different data structures, and with that I think I'm running out of time.
2 or 3 minutes for questions.
Yes, yeah.
Oh, but do you have any final statement?
Basically, I can just run over the things I intended to say. There's the approach, that we're taking from the biologist's point of view is sort of complementary to a lot of what we've been talking about this week here, and that based on the framework of Kendall shape space, which is a solid and manageable basis for looking at the complexity of analysis is in the data structure, and the analyses we're doing in order to answer some quite specific questions that come out of biology. I think there's a lot of potential for further discussion, but I think that there will have to be a lot of discussion to bridge the differences in emphasis between the disciplines. Thank you.
So I have this question for a long time. There seem to be only 600,000 species of plants, but at least 2 million species of beetle. Why is that? Is that the question of taxonomy, or are there more evolutionary niches for plants, less evolutionary niches for plants than for animals? And are there more animal-eating animals than plant-eating animals? I mean, this is a question for statistics of evolution.
Yes, that's a very good question, and a very, very hard question. Also, actually, most of the relatively few plants have evolved quite recently. So for most of the history of life, it has been very animal heavy. And actually, probably both on the animal and the plant side, the interaction between animal and plants is very important. So a lot of the differentiation in plants is about interaction with pollinators and dispersers, and a lot of the beetles are eating different kinds of plants or that. There is, I guess you can argue in a somewhat speculative way that all plants are making their living in a relatively similar way. They're growing in a particular, either in the soil as land plants or floating in the water or attached to the thing as aquatic plants, and they're photosynthesizing. Whereas in animals, there's a much bigger range of those that eat plants, those that eat other animals in different kinds, those that eat organic matter, leftover, dead organic matter. So there might be more niches there for animals than plants.
Plus animals can move. So I think we'll
on that question.
you want to follow up? Okay.
Yep. So plants are also very plastic, or in the sense that they adapt easily to their environment, or they have more adaptation to the environment. So one species can live in different more environments than one species of having itself.
Yes. That's actually something that's related, that shows up in shape analyses. And my limited experience with plants, plants tend to be more variable within species, substantially more variable than most animals. So that's
Thank you very much, Chris.
Thank you.
It's very interesting.